Imagine you've just woken up from the egg retrieval procedure.
Right, you're still in recovery.
Exactly. You're in recovery, and the doctor walks in, smiling, and says, "We collected 10 eggs."
Which is an amazing feeling.
It really is. I mean,
Oh.
for you lying there after weeks of injections and, endless ultrasounds, it feels like the finish line.
You finally get to just exhale.
Yeah, you take a breath. But down the hall in the quiet, completely temperature-controlled environment of the embryology lab, the clock has actually just started ticking.
It really has.
Because those 10 eggs are not a finish line at all. They are, well, they're the starting line.
Very much so.
And that's why we are continuing our journey through our Inside the IVF Lab series today. And we are intentionally slowing down the pace for this one.
We really need to.
Yeah, we want to match that meticulous, deliberate, almost meditative work happening right now under the microscope.
I love that we're doing this because in our previous discussion, we focused entirely on the physical act of finding the oocyte within the follicular fluid.
Right, the treasure hunt.
Exactly. But simply pulling a cell out of an ovary does not mean it is actually ready to create a life.
Right.
So our mission for this deep dive is to shift our focus entirely. We are moving from simply finding the oocyte to, well, truly reading it.
Reading it. I like that.
Yeah. We're going to explore how an embryologist assesses the maturity of that single cell, what the physical shape and structure actually reveal, and
I think this is the big one.
yes, perhaps most importantly, we have to confront the humbling limits of what human eyes and microscope lenses can actually predict.
It's a lot to cover, so I want to start right at that moment after retrieval. The fluid comes into the lab, the embryologist spots the egg. But what are they actually looking at?
That's a great question.
Because from what I gather about this process, a fresh oocyte isn't just sitting there fully exposed like, I don't know, a pearl on a table.
Far from it. When an oocyte is aspirated from the follicle, it is a massive, highly specialized cell, but it is completely encased in a dense protective cloud of supporting cells. Yeah, a cloud. To understand what the embryologist is eventually trying to evaluate, we need a clear picture of the anatomy here.
Okay, walk us through it.
So at the very center, you have the ooplasm, or the cytoplasm, that is the core biological material of the egg.
The actual, like the yolk of the egg, so to speak?
Sort of, yes.
Mm-hmm.
And surrounding that core is a tiny fluid-filled gap called the paravitaline space.
Paravitaline space, got it.
Right. Just outside of that space is a thick, protective outer shell called the zona pellucida.
The shell, okay.
And then clinging tightly to the outside of that shell, radiating outward in all these layers, are the cumulus and corona cells.
Wow, so there's a lot going on there.
There really is.
Yeah.
These are the cells that fed and communicated with the egg while it was developing inside the ovary.
Wait, so it's not a bare cell at all when it arrives in the lab? Okay, let's unpack this. It sounds almost like it's an incredibly fragile item that was shipped inside, like layers and layers of cellular bubble wrap.
That is a highly accurate way to visualize it, honestly.
Because you know there is something valuable at the center, but you can't actually see its condition because of all the packaging.
Exactly. The embryologist is looking at this fuzzy, expanding mass under the microscope.
Just a big fuzzy blob.
Right. And if the lab is preparing to do ICSI intracytoplasmic sperm injection,
Where they select the single sperm and inject it directly into the egg, right?
yes, exactly. They cannot do that through all those layers of cumulus cells.
Yeah.
It's just not possible.
They'd never be able to see what they were doing.
No. So they have to perform a procedure called denudation, usually a few hours after the retrieval.
Denudation. Okay, so how do they actually get all that bubble wrap off without damaging the oocyte inside?
Very, very carefully.
I mean, I imagine you can't just scrape it off.
You certainly cannot. Denudation relies on a precise combination of chemistry and physics.
Okay, chemistry first.
Right. The cumulus cells are held together by this sticky, gel-like matrix made largely of hyaluronic acid.
Which sounds familiar, like from skincare.
Yes, exactly the same stuff. So the very first step is to briefly expose the egg to an enzymatic media containing hyaluronidase.
Hyaluronidase, so an enzyme that breaks it down.
Exactly. This enzyme acts like chemical scissors. It specifically targets and dissolves that hyaluronic acid, breaking the bonds between the outer cells.
Okay, I see.
It's like dissolving the mortar between bricks, so the whole structure just loosens up.
But the enzyme doesn't do all the work on its own, right? You mentioned physics, too.
No, the enzyme only loosens the outermost layers. After a very brief, carefully timed exposure to it, the embryologist moves the egg to a clean culture drop and begins the mechanical phase.
The physics part.
Yes. They use a finely pulled glass pipette, which has an opening just slightly larger than the oocyte itself.
Just barely bigger.
Right. By very gently drawing the egg up into the pipette and pushing it back out, the sheer force of the fluid strips away the remaining corona cells hugging the shell.
Oh, wow. I would be absolutely terrified of applying too much pressure.
It is stressful.
The sheer force alone sounds really risky. I mean, is there a real danger of damaging the oocyte during this step?
Absolutely. Overhandling, pipetting too vigorously, or using a pipette that is just slightly too tight can completely fracture the zona pellucida,
Oh,
or
no.
it can fatally damage the cytoplasm inside. The embryologist must maintain an incredibly steady hand and just total focus.
It's high stakes right out of the gate.
It really is. It is a vital, transformative moment. The denudation strips away the veil, making the oocyte fully readable.
For the first time.
Exactly. I should note, though, in conventional IVF, where sperm and egg are just left to fertilize naturally in a dish, full denudation isn't usually done up front
Really? Why not?
because those cumulus cells actually help facilitate the natural sperm binding process. But for ICSI, the veil has to come off so the lab can plan the next step.
Okay, so the bubble wrap is gone. We have a naked, fully visible oocyte sitting under the microscope. What is the very first thing the embryologist is looking for?
The immediate overriding priority is establishing the stage of nuclear maturity.
Nuclear maturity, meaning what's happening with the DNA.
Precisely. An egg cannot be fertilized by a sperm if its genetic material is not in the exact precise configuration.
Right.
Human cells typically have 46 chromosomes. But a reproductive cell, an egg or a sperm, needs to have exactly 23.
So that when they combine, the resulting embryo has the correct 46.
Exactly. And the biological process of having those chromosomes is called meiosis.
Meiosis. And the embryologist can actually see where the egg is in that meiosis process just by, looking at it.
They can. They categorize it into 3 distinct stages based on visual markers.
Okay, let's go through.
The first stage is the GV, or germinal vesicle stage. This is a profoundly immature egg.
Okay, what does GV look like?
Under the microscope, inside the cytoplasm, the embryologist can clearly see the nucleus. It looks like a distinct intact circle or vesicle.
Just a circle floating inside.
Right. And seeing that intact circle means the egg's chromosomes are still all gathered together inside the nucleus. Meiosis has not fully resumed.
So it's essentially paused in time.
Very much so.
But wait, why would it be paused if the patient was just triggered to grow these eggs by the stimulation medications?
It's a tricky biological quirk. In the natural cycle, eggs wait in this paused GV state until a massive hormonal surge just before ovulation tells them to resume dividing.
Right, the trigger.
Yeah, but sometimes, despite the medication triggers given before retrieval, an egg just doesn't get the message.
Oh, wow.
Or it just doesn't have the internal machinery to respond in time, so it comes out as a GV.
Got it. So GV means a hard pause. What happens if it has started moving forward but isn't quite done?
That brings us to the second stage, M1, or metaphase 1.
M1.
At this stage, the egg has resumed meiosis. The germinal vesicle breaks down, so the embryologist no longer sees that clear circle inside the cytoplasm.
The circle disappears.
Right. The chromosomes have started to align and separate. However, the egg still hasn't completed the final step of the division.
So what are you seeing exactly?
Visually, there is no nucleus, but there is also no exterior sign that the chromosomes have successfully halved.
It's in transit. Moving, but just, not there yet.
Exactly.
I assume neither GV nor M1 eggs are used for ICSI then, because they aren't ready.
For standard routine ICSI, no, they are not used.
Makes sense.
While some might mature a few hours later in the incubator, injecting them while they are immature is generally associated with failed fertilization or highly abnormal embryos.
You just don't want to force it.
Right. The lab is searching for the final stage,
Hmm.
M2 or metaphase 2. This is the mature stage.
Okay, so what does an M2 look like compared to an M1? How do you actually know it finished the halving process?
At the M2 stage, the embryologist will look into the paravitalized space.
Wait, that's the tiny fluid moat between the core of the egg and its outer shell, right?
Exactly, the moat.
Yeah.
There, they will see a small, distinct spherical structure sitting right next to the egg. That is called the first polar body.
A polar body. What exactly is that? Sounds mysterious.
It's actually really practical. When the egg divides to have its chromosomes, it doesn't split into 2 equal cells like other cells do. Because the egg needs to keep almost all of the cytoplasm, all the nutrients and all the cellular machinery for itself to support the future embryo.
Ah, so it hoards everything.
It absolutely hoards it. So it packages the 23 discarded chromosomes into a tiny, tightly wrapped bundle and pushes it completely out of its own cytoplasm into that moat space.
Wow.
That tiny bundle is the polar body. It is essentially the biological trash can for the unneeded genetic material.
That is amazing. So seeing that biological trash can sitting outside the egg is basically the ultimate green light.
Exactly.
It tells the embryologist, the chromosomes have been halved, the trash has been taken out, we are ready for sperm.
What's fascinating here is that this visual confirmation of the polar body means the egg has completed the key physical maturation step.
It crossed the finish line.
Yes. Identifying M2 maturity is the first major, uncompromising filter between egg retrieval and embryo creation.
You know, this really circles back to the clinical reality we opened with.
You woke up, you heard 10 eggs, and you felt this wave of relief. But if the embryologist strips off the cumulus cells and finds that, say, 3 are GVs and 1 is an M1,
Then your starting number for fertilization isn't 10.
it is suddenly 6.
Right.
And that drop-off, I mean, that can be incredibly jarring for someone going through this process.
Yeah.
You feel like you lost 4 eggs.
It is very tough news for patients, but it is a strict biological necessity. The lab has to find the M2s.
Let's focus on those 6 remaining M2 eggs then. The green light is on for all of them. Are they all considered entirely equal at this point?
Definitely not.
Like, does the lab just line them up for sperm injection, or is there another level of assessment?
Oh, the lab definitely does not stop looking. Beyond the stage of nuclear maturity, the embryologist must assess the physical architecture, the overall morphology of the egg.
The morphology.
Right. The running joke in the field is that this is the microscopic beauty contest.
It's beauty contest. Okay.
Once an egg is identified as M2, it becomes immediately clear that not all M2 eggs look the same. So what makes an egg beautiful to an embryologist?
A textbook, visually perfect oocyte is perfectly spherical.
Like a perfect globe.
Yes. The outer shell, the zona pellucida, is a uniform thickness. The paravitline space is clear and narrow.
No clutter in the moat.
Exactly. The cytoplasm inside is smooth and evenly textured, and the polar body is a neat, intact little circle.
Sounds pristine.
It is. But biology rarely produces only textbook examples. Embryologists are trained to scan for visible deviations from this ideal, which we call dysmorphisms.
Dysmorphisms. That sounds intensely clinical. What kind of deviations are we actually talking about here?
Well, they look at the cytoplasm first. Instead of being smooth, is it dark or heavily granular, like it's filled with fine sand?
Okay, sandy texture.
Right. They look for vacuoles,
Mm.
which are small fluid-filled pockets, or bubbles right in the middle of the cell.
Bubbles inside the egg.
Yeah. They check for clusters of smooth endoplasmic reticulum, which look like slightly darker flat discs. They also look outside the core. Is there cellular debris floating around in the paravitline space? Is the zona pellucida unusually thick or strangely thin?
The structural stuff.
And they evaluate the overall shape. An oocyte might not be spherical at all. It might be distinctly oval or irregular.
So what does this all mean? Hold on. If the egg has fluid pockets inside it and a granular sandy texture and it's shaped like an oval,
I know where you're going with this.
I mean, how is it clinically justifiable to still use it? Why wouldn't the embryologist just throw an egg like that away to be safe? It sounds like it's fundamentally defective.
That instinct is incredibly common, and it brings us to one of the most pervasive and consequential myths in reproductive medicine.
Which is?
An ugly egg is not automatically a bad egg. Morphology provides us with clues, but it does not provide absolute certainties.
But if it looks abnormal, doesn't that inherently mean the machinery inside is broken?
Not necessarily. Different laboratories have different protocols. Some document every single vacuole and granule in minute detail, while others only record severe abnormalities. Right.
But the clinical consensus across the entire field is that the predictive value of an egg's appearance is notoriously inconsistent.
Inconsistent how?
We have data showing that an unusual-looking oocyte one with a granular texture or a distinct oval shape can absolutely fertilize normally, develop into a robust, beautiful blastocyst, and result in a perfectly healthy pregnancy and baby.
Wait, really? That is wild to me. The visual defects don't inherently stop the biology from working?
They really don't. Conversely, an oocyte that looks like a flawless textbook sphere with perfectly smooth cytoplasm and an ideal polar body might completely fail to fertilize.
Just do nothing.
Or it might fertilize and then just arrest, stop growing completely on day 3.
And that implies we are missing something massive.
We absolutely are.
If an egg can look absolutely picture perfect, it took out the genetic trash, it is beautifully spherical, and it still stalls out, what is actually happening?
It's the big question.
Here's where it gets really interesting. I keep thinking of this like buying a used car.
Okay, I like this. Reaching the M2 stage, having that polar body, means the car looks fully built on the outside.
Right.
It has 4 tires, a steering wheel, the doors are attached. The morphology, the beauty contest is just the paint job.
Exactly.
Maybe there's a scratch on the door, maybe the color is faded, that is a dysmorphism. But none of that visual information tells you what happens when you sit in the driver's seat and turn the key.
That is so true.
The real question is, does the engine start? Does the transmission actually work?
Your car analogy perfectly illustrates the profound dividing line between what we call nuclear maturity and cytoplasmic competence.
Cytoplasmic competence.
Yes. Nuclear maturity is having that polar body. The chromosomes are right, the physical structure is there, we can see it.
The car has tires.
Right.
Yeah.
But cytoplasmic competence is the internal functional readiness of the egg to actually support fertilization and drive the complex development of an early embryo.
So cytoplasmic competence is the invisible engine under the hood.
Exactly. And routine microscopy simply cannot assess that engine.
Because it's too small.
Yes. Cytoplasmic maturation involves incredibly complex molecular processes. It is about the organelle-level readiness.
The tiny parts inside the cell.
Right. For instance, think about the mitochondria, which are the powerhouses or batteries of the cell.
Right, from high school biology.
Are those batteries fully charged and distributed correctly to provide the massive amounts of energy required for cell division?
And you can't see the mitochondria through the microscope.
No, they're far too small. The engine also relies on mRNA transcripts.
What do those do?
When the egg is developing, the mother's body loads it with specific RNA and proteins. These act as the instruction manuals for the embryo.
Like the software.
Yes, because for the first few days after fertilization, the embryo hasn't turned on its own DNA yet. It is completely running on the software left behind by the mother in the cytoplasm.
Oh, wow. I had no idea.
It's amazing. If those mRNA transcripts are degraded or the proteins are misaligned, the cell will simply stop dividing, no matter how perfect it looked from the outside.
The microscopic machinery is basically locked inside a black box.
It really is.
So if we can't see the engine, what actually dictates whether an egg has that cytoplasmic competence? Why does one egg have a perfectly charged battery and flawless software while another one just stalls out?
If we connect this to the bigger picture, it is driven by a web of interconnected factors. Maternal age is one of the most critical.
Because things just slow down.
Yes. As a woman ages, the molecular machinery inside her eggs naturally becomes less efficient. The mitochondria degrade, meaning the egg literally produces less energy.
The batteries hold less charge.
Precisely. The chromosomal spindles become more fragile. The body's response to ovarian stimulation medications also plays a role, as does the highly localized environment of the individual follicle where that specific egg grew.
So the environment in the right ovary might be slightly different than the left, impacting the eggs differently?
Even more localized than that. 2 follicles sitting right next to each other in the same ovary can provide totally different environments.
That is wild.
And that brings us to the reality of intrinsic biologic variability. Even within a single patient's cohort of eggs, retrieved on the exact same morning, the variation is staggering.
Because they each had their own little micro-environment.
Exactly. You might retrieve 10 eggs, 6 are M2s, 4 look perfect, 2 are oval. But their internal cytoplasmic competence will vary completely independently of all that.
Independent of the looks.
Right. A single retrieval always produces a mixed population of eggs with wildly different developmental potentials. The lab can identify the physical maturity, but the ultimate competence of that cell remains completely hidden until we actually introduce the sperm and observe what happens.
It really changes the entire perspective of what an embryologist is actually doing back there.
It requires a lot of humility.
I bet. We've spent this deep dive slowing down to understand this process. We started with a cloud of cells and explored the delicate, almost surgical process of denudation.
Using chemical scissors and physical shear force to strip away the bubble wrap.
Right. And then we broke down how embryologists read the nuclear maturity, tracking the egg from the pause GV stage through M1 to the crucial M2 stage where it extrudes that polar body.
Taking out the genetic trash.
Exactly. And we reckoned with the limits of the beauty contest. We established that an ugly egg with vacuoles or granular cytoplasm can still hold a perfectly functional molecular engine, while a beautiful egg might lack the energy to divide.
Because morphology gives us context, but cytoplasmic competence is the biological engine that actually drives life forward.
Which leaves us with a final, lingering question for you to think about.
The really big one.
Right now, the true developmental potential of the egg, that molecular engine, the mitochondria, the mRNA, is mostly invisible to us. We have to guess at the interior by looking at the exterior. But, science doesn't stand still.
Never.
How might technology eventually bridge this gap? Could we develop non-invasive ways to read the metabolic signature of an egg in the culture media without ever harming it?
Like a breathalyzer for an egg.
Yeah, exactly. Or could artificial intelligence be trained to identify subtle, microscopic visual patterns of cytoplasmic competence that the human eye completely misses? The way we read the oocyte today might look incredibly primitive a decade from now.
The field is aggressively pursuing those exact technologies. It's a fascinating frontier.
It really is.
But for now, the meticulous work under the microscope continues. Thank you for slowing down with us today. In our next deep dive, we're going to shift our focus entirely to the other half of the fertilization equation.
The sperm.
Exactly. We will step into the andrology workflow to explore sperm preparation. We will cover how the lob uses density gradients and the swim-up technique to isolate the best cells, and exactly how sperm is selected for conventional IVF versus ICSI.
The oocyte has been read and is waiting in the incubator. Now it's time to prepare the sperm. Until next time, keep diving deep.